So good afternoon, everyone, and thanks for joining us. Welcome again to our 2023 NECAN webinar series highlighting monitoring priorities in the Northeast. This is our 10th webinar in this series, and today's theme is New Tech and Sensors Part 2. With the assistance of this series and our presenters, the NECAN Steering Committee will be working on the development of a regional monitoring plan for ocean acidification. And these webinars will serve as a resource for them as the plan begins to come together. Updates on this series are shared through our mailing list and on our website, so be sure to check out our website for the full schedule. At the conclusion of presentations today, the Steering Committee will be asking our panel some questions, and then we're going to open it up for a more general Q&A. And during that Q&A session, please feel free to use the raise hand function on the bottom of your screen, or you can submit questions and comments in the chat, and we will, the facilitator will get to them. Today we have three wonderful presenters, Jamie Palter, Mike Brosnahan, and Adam Subhas. And first up, we'll hear from Jamie. Jamie is an associate professor of oceanography at the University of Rhode Island. She studies ocean circulation and biogeochemistry, focusing on the transport of heat, carbon, and oxygen through the ocean and across the air-sea interface. To do this, she collects new observations with moored and autonomous platforms, including sail drones and sea gliders, and combines them with vast archives of historical, hydrographic, and satellite data. Her work has often focused on the Gulf Stream, including its influence on climate and its role in the overturning circulation. She has recently become involved with the scientific assessment of marine carbon dioxide removal through enhancing ocean alkalinity, which has led her to pursue projects at smaller scales and closer to the coastline. Jamie, feel free to share your slides when you're ready.
Thank you for that nice introduction. And let me see, we didn't have a chance to practice ahead, so let's see if this works. All good on the share screen?
Yes, I see that perfectly. Thanks, Jamie.
Okay, great. Thanks for the invitation, which came from Sam Sidlecki back in January or something. So, it was a little, and I'm excited to talk to this group today about my work, which I focused on my work with sail drone. And with that, I'll just jump into what I prepared. So I was told that this was not to be a normal science talk, that I had a certain assignment kind of to assist NECAN as it develops its regional ocean acidification monitoring plan with a focus on new tech methods and sensors to address these questions about new technology helping with ocean acidification monitoring, whether this new tech would replace, augment, or add new capabilities, and to think through what are its readiness, the quality of the observations, what expertise is needed to run it, and how much it costs. So I'm going to try to run through that. I have not really been an active player in the ocean acidification world, but I've been using sail drone uncrewed surface vehicles, these autonomous surface platforms, to measure ocean PCO2, which is obviously highly complementary. I also use gliders, an underwater drone that can incorporate pH sensors, but I don't have one, and most people don't. So we can talk about that a little bit. The gliders operate very much like Argo floats and can hypothetically carry a pH sensor, but there's very few people that actually have these sensors built into their sail drones. And with supply chain issues with the pH sensors, like the FET style pH sensor, due to Honeywell's, I don't know how many people are aware of this, but Honeywell hasn't been making the chip. These are really hard to come by. Even BGC Argo has been struggling, although has a workaround. And so I'm not really going to talk about gliders because I just don't feel like the readiness is close for that. There's another group in Alaska trying to make a PCO2 enabled glider, but they're also in prototyping and development stage. And so finally, I'll just thought I would use the platform a little bit to consider expanding the vision of our ocean acidification research to include baselining for marine carbon dioxide removal research. Okay, that's I hope not too long a preamble. And I said I wouldn't do a normal research talk, but I did want to just give the context in which I've been taking these observations. I've always conceptualized the research that I've done in this space around narrowing the uncertainty on ocean carbon uptake, although that has implications for pH and acidification. And this plot has been used many times to motivate this research. It shows the estimates of global ocean carbon uptake through the years from 1960 to present, and the data-derived products in the lighter green and the ocean model derived products in the dark green. And these just don't agree to within about 4 gigatons of ocean carbon uptake. And that difference, instead of narrowing over time as the science has gotten better, it's expanded. And that difference of those 4 gigatons is larger than the sum of all pledged avoided emissions in 2030. So people will say, we really won't have a geophysical check on whether we've achieved those goals set forward in the Paris Climate Accord, because we can't close the carbon budget to that level of certainty. And of course, regional and local uncertainty is much, much higher than global. So one of the sources of that uncertainty is the lack of surface ocean PCO2 measurements. And that's because it's principally measured with ships and buoys, and there's huge gaps. This is the SOCAT's Surface Ocean CO2 Atlas, with all the CO2 observations since 1957, and it looks really well sampled, mostly on these ship-based platforms, and to some degree on the like the moored platforms shown in squares. It looks really well sampled, but if we go to any given year, like 2019, there's huge gaps all over. And then it still looks like our Northeast is well sampled, so we've done a much better job. But if we look at the winter time, we'll see it's still very poorly sampled, and I'll show that in the next slide. So it begs for new capability for long duration, high resolution ocean atmosphere observing, even under challenging conditions. So let me just show you what the winter time looks like. This is 2018, December to March of 2018, 2019. And the 2018 winter in the Southern Hemisphere, essentially no measurements in the Southern Hemisphere. We still have some in our Northeast region, and you might be able to make out one squiggle. That one squiggle was the sail drone. It was essentially the one of 2 observations made in this region in winter of 2019. So we were out with a sail drone in January 2019 and occupied this seemingly random spaghetti trajectory, but what we were doing was trying to crisscross the Gulf Stream as we move downstream. Okay, so that is an intro to say why I've been using sail drone, but let me tell you a little bit more about them. They are called uncrewed surface vehicles, and they are, this little guy here is shown for scale, so that's about a 6-foot human being relative to the size of the platform. They carry an entire meteorological package above the water, so relative humidity, wind speed, anemometer for wind speed, sea level pressure, all of those things, surface air temperature. They can carry additional sensors. They can carry a huge payload because they charge all of the science sensors with solar panels. You can see one here in this picture, and I have a picture of my graduate student, Sarah Nickford, who worked up a lot of the data that you'll see coming up. Then in the water on the keel, and they carry an entire suite of hydrographic sensors, Seabird CTD, and under an oxygen sensor. And sometimes they carry 2, they've incorporated Seabird and under oxygen sensors. And most importantly for our work has been the incorporation of NOAA's ASV CO2 sensor. This was developed at PMEL as the MAP CO2 sensor for incorporation on the Tau moorings in the tropical Pacific. They had great success with that and did a lot of adaptation to make it ready for ASV, for deployment on these, they're calling them ASVs, autonomous surface vehicles now, for incorporation on there. And what's great about this is it has a way to sample in the air and in the ocean at the same time. So if you want to get fluxes you need that delta, the air-sea difference in PCO2, and you have both of those things here, plus wind speed, the other piece that you need, and SST and sea surface salinity. Okay, so I think because this is focused on sensors and technology, I wanted to give you a behind the scenes view of the ASV CO2 sensor with the full recognition that I came to this as a user, and I've, had the privilege of working with Adrienne Sutton, who was one of the developers. She worked with Chris Sabine at PML to do this, but to tell you a little bit about what it does. It pulls in a sample from the air and a sample from the ocean, but before it measures that sample, it performs a 2-point calibration with every step. It measures a 0 by scrubbing the CO2 from the sample by bubbling it through soda lime. It also carries on board a WMO reference gas, certified reference material. And so before each measurement, it does a 0 and it measures its span gas to get a 2-point calibration and for the sample itself. The entire cycle takes about 17 minutes. So it's a limitation on how frequently you can make these measurements. Typically, we sample at 1 hour because it's also very power hungry, but you can go to half hour without compromising anything about that cycle. Ultimately, it produces a product that's within 2 microatmospheres of accuracy. That's like the nominal accuracy, but we found that it's about right. Early versions of the ASV CO2, the one made at PMEL, was a full system and it incorporated a pH sensor that was highly complementary. Saildrone now produces this system in-house, and they've tried to simplify it to a single sensor, and has removed the pH. That was, from our point of view, was a real loss because although they didn't provide fully independent information, they were very complementary. The pH drifted, but was not noisy under very strong wind and waves. And PCO2 was the reverse. The PCO2 was extremely stable owing to its 2-point calibration, but sometimes would suck in what we think was a mixture of air and water under very high sea state. And so it would be noisy. So with pH, we could remove the noise. And with PCO2, we could remove the noise in the PCO2. And with that noise-free PCO2 record, we could remove the drift in the pH. So we would like to see that come back, the paired capability. To look at the kind of results we had, this came from Sarah's 2022 paper. So in that deployment, we had 18 days of Saildrone data before the Saildrone was damaged by very rough sea state. We crossed the Gulf Stream 5 times. Which is as much as happens under like the best of circumstances in an 18-day research vessel cruise, and measured CO2, air CO2 every hour over those 18 days. So this is like a step change in the amount of data you can collect under these conditions at this price. And I'll get into price in a minute. The colored trace is the sea surface PCO2 as measured by the Saildrone. The black trace is what the Landshutzer product would, using a neural net procedure to estimate PCO2 from predictor variables collected by satellite, would give you. And I think the Landshutzer product is absolutely remarkable. It looks really good, and yet it's a monthly product at 1 degree resolution. It's just not made to see the kinds of gradients we're seeing. The red colors are in the Gulf Stream itself. Oh no, are south of the Gulf Stream. The yellow colors are in the Gulf Stream itself, and the blue colors are north of the Gulf Stream. And so this is just a super high gradient region. You can see over the course of and this is wintertime. So there's lots of water masses overturning all of these things. And so you can see there's up to 50 microatmosphere difference between the lowest and highest days that we measured. Thinking about, again, my assignment in this Northeast, the NECAN assignment was to think through how we could use this for ocean acidification research. I just wanted to put this out here that PCO2 and pH collect highly complementary or very, very similar data. So if you only had PCO2 and you could pair it with the temperature salinity, the salinity alkalinity relationship, you could deduce pH. I don't know if that's at the accuracy that the network would want, but that relationship is very tight, and the PCO2 data is very highly accurate. So it may offer a road forward. Just put that out there. Okay. I just showed you the data from our 2019 mission. We were also out there in 2021, 2022. And this time with 3 sail drones almost for the entire winter. And so this is just my favorite animation that actually Sarah worked with our summer student, Julian Lin, to make. He's a visiting summer student. And this is the turbulent heat flux for December and January 2021-22, and the C3 sail drone pass over it. And I know this doesn't have a lot to do with ocean acidification, but the reason I want to show it is because there's such rich structure here. So most atmospheric scientists would imagine that the atmosphere and its cold storms in winter blows a huge storm over the ocean, sucking heat out of the ocean, and due to these storms, and that the atmosphere exerts the biggest control. And that's mostly true. All of these big bright colors that you see, up to 1,000 watts per meter squared of turbulent heat flux, which is a crazy number. It's like 2 or 3 times the heat of the sun, more heat coming from below the sail drone than above the sail drone. Those big boluses of heat flux are due to the storms. But underneath, there's this rich patchwork, of gradients due to the warm Gulf Stream that the storm blows over. And all of that mesoscale variability under the storm influences the storm itself. It also influences the ocean response. And that the same would be true for all of the biogeochemistry. And so we're trying to get at some of that variability, the mesoscale variability in these air-sea interactions. Okay. So I have another postdoc that was working on this. He's since moved on to PMEL, actually, to NOAA. He trained a neural net of his own using the sail drone data and a bunch of first with sail drone data only, and then with satellite data interpolated along the sail drone path. And so I asked him to compare this to the Landshutzer product that's in black to have a view similar to what Sarah did. And he was nice enough to whip this up in a jiffy for this talk. But we had been working on the neural net, for a long time before that. And so the blue now is the true observed sail drone just in December. And this is because Landshutzer isn't updated to 2022. It's just through December 2021 for the 3 different sail drones. The neural net is what he's made from the satellite data. And you can see that it matches quite well. The black is Landshutzer. Again, this is not to criticize Landshutzer. It's a remarkable product, but these global coarse resolution 1 degree and monthly products can't get this kind of very high resolution view. The goal ultimately is to take this really accurate picture and highly resolved picture of PCO2 at the ocean surface and turn it into maps like the map that you saw in the previous slide for heat fluxes. And we could do the same for carbon CO2 fluxes and get a more accurate carbon flux for the North Atlantic and more accurate surface ocean PCO2, and with implications for pH as well. So this is kind of the promise of having this highly resolved, very accurate data persistent over a seasonal cycle. Okay. Getting to the particular questions that you've posed is the readiness of these platforms. Fleets have been deployed in almost all conditions. There have been notable successes. Sail drones circumnavigated Antarctica. We had full winter persistence in the Gulf Stream. There's been a mission to the Arctic right up against the sea ice. But there have been many challenges. That list really hides the fact that the Antarctic mission used a shorter wing that could persist in very strong storms. But that shorter wing is not as maneuverable. So it went downwind the whole time. An ACC mission is perfect for that because in the westerlies, it can just go around and around and make a full Antarctic circumnavigation. That's not good for the Gulf Stream. So we couldn't use the shorter wing in the Gulf Stream, but the Gulf Stream's waves are nearly as big, not quite, but nearly as big as the waves in the ACC. And of course, we did damage over the course of the 2019 and 2022 missions. We damaged 3 sail drone wings and high waves. So this is something to keep in mind. When the waves are taller than the wing by a bit, it can basically turtle the sail drone and snap its wing. There can be hazards in highly trafficked waters. These are autonomous vehicles. The Coast Guard has to approve of their use. So I don't know if that's the best for a coastal application, but surely that's a question for you all. And then there's real challenges in powering the instruments in wintertime cloudy conditions. Those solar panels are not that big. And when there's low sun angle and lots of cloud cover, as we experienced, you just can't always power the sensors. We had 3 sail drones in the Gulf Stream in February, and 2 of them were off. So that was a real disappointment. 2 of them are just for 2 weeks, but 2 weeks in the middle of our mission, it was really painful. Then turning to the ASV CO2 sensor, which is the one that I think has the most relevance here. This is a really high quality sensor or sensor system. We have compared it as much as possible between sail drones, flying them in tight formation. They're super precise. We have flown by Pro-Oceana sensors on the Ocean Observing Network, the OCA Pioneer Array, very close. We even put a bucket into the water. Hongji Wang led this, put a bucket into the water when we were kind of in touching distance of the sail drone to collect DIC and compare that with the DIC deduced from sail drone. Also quite accurate, but a very hard comparison to make. There have been some supply chain issues with a valve component. They used a different valve in the latest generation, and we had some failures related to that valving. And again, the loss of the pH from the system we felt was a real loss. Cost. I feel the cost in the sail drone model is data as a service. So sail drones are not for sale. They're deployed and piloted by saildrone.com, and their data is transmitted via Iridium and served via FTP server to the PI. All of that's done by saildrone.com. So I say that using a sail drone, 2 minutes, yeah. Using a sail drone is like hiring a landscaper. You don't own the equipment and you're not the guy on top, but you tell him where to go and what to do, right? This is not the only model for USVs. Wave Glider are bought and sold. PIs own them. They deploy them. They have a technician usually in-house to deal with them. This is the Wave Glider. So that's like owning your own lawnmower. I like the data as a service model because the pilots are expert mariners. They can the magic that they have with these sail drones, they will fly them in a gale from hundreds of kilometers apart, and they are almost touching. If you want to do an intercomparison, it's really amazing. The company also assumes much of the risk. I have now in my work damaged 3 sail drones that has never come out of any budget of my own. That was their risk that they assumed. Not happily, and they do try to avoid those risky situations, which can change your deployment strategy. There's no need for an in-house technician, which is very hard to support, although you don't get to have that hands-on piece. The cost is not low. They are, when I was using them, I don't know if they've gotten any different, it was $2,500 per sail drone day, with the idea that you would deploy at least 2 at a time. So $5,000 per day if you have a 2 sail drone deployment. That's a lot of money. Expertise needed. The appeal of data as a service means that the scientists can focus on science. So and I think that's a strength. You just work with the pilots and mission managers to design waypoints and a sampling plan. For us, we monitored at least daily. We were looking at it sometimes many times a day, depending on where it was and what the storm conditions are. I don't know if that would be necessary under more standardized monitoring strategies. And then how you want to have enough expertise to think about calibration, quality control, and quality assurance. Like, are you comfortable going with the newly calibrated Seabird CTD, whatever oxygen sensor, ASV CO2, or are you going to want to do on the fly and in the field calibrations? We can talk through that if there's time for Q&A. And that's all I prepared today. I didn't think it would take that long, but thanks for sticking with me, and I'm happy to answer questions.
Thanks, Jamie. That was an amazing presentation. We're going to do like a general Q&A all together at the end. So I'm sure there will be questions from the steering committee, but for now, we're going to move along to Mike Brosnahan. Mike is an associate scientist at the biology department at Woods Hole Oceanographic Institute. He studies harmful algal bloom, or HAB, ecology and dynamics through the development of novel in situ observing systems. A major focus of his work with Syracuse and other Gulf of Maine-based partners has been the establishment of Habon New England, a network of advanced phytoplankton sensors for improved surveillance and monitoring of HABs. The system aims to make sophisticated HAB-related data and products available to resource managers and stakeholders in real time, while also facilitating new types of scientific investigations for the better understanding of how HABs are responding to climate change across the globe. Please feel free to share your slides, Mike.
Thanks, Austin. Let's see. I might need a little bit of coaching on this one. We'll see how this works. Are you seeing, what are you seeing right now?
I see your slides full screen, and I was seeing a video playing as well, which looked great.
Okay, so you're just seeing the slides though.
Yes.
Okay, great. Yeah, so thanks. I just, as I was putting this one together, I saw this amazing set of graphics that the Washington Post compiled in an article last week. And I think, my presentation today, I'll be talking about ocean acidification and HABs as two interrelated factors that are being driven in large part by warming of the ocean. And this really illustrates well what a hotspot our region is for warming. So that in terms of the topics I want to tackle today, this sort of framing of HABs as the fourth horseman of the climate apocalypse. This is something that my colleague Chris Gobler proposed some years ago. And I'm a HAB person, so of course I'm pretty partial to it, but I think it has a lot of merit. And then I'll tell you a little bit about this HAB-on-NE sensor network that we've been working to establish with NERACUS and other partners. And then I just want to introduce a new project that we've just started that's really looking at potential interaction between acidified waters in coastal zones and the development of HAB events. So first, what are HABs? This is a really diverse group of organisms, of algal organisms, so cells in the ocean that do photosynthesis that can cause a wide range of negative societal impacts. So that can range from really productive blooms that generate just tons of biomass that by itself is a nuisance, but also when it degrades can cause really severe hypoxia and other ecosystem degradation. In freshwater systems, there's a range of organisms that can contaminate drinking water supplies and impact aquatic life. And then in my group, we're particularly focused on cells that produce biotoxins and can either poison seafood or otherwise negatively impact seafood production. And so why is this the fourth horseman? Well, this isn't mine, but we have a range of impacts on the ocean that we recognize that are related to climate change. And first among these is warming. And so this is just another illustration, a really beautiful one that is available through the Climate Change Institute at the University of Maine that shows just how dramatic the warming we are experiencing in our region is. So if you look at that plot, there's the multi-decadal average and its confidence interval in the dashed lines. 2022 is in orange, and you can see it was quite a warm year. And then in the dark black, the solid black line is this year, and you can see we're well outside the envelope of what we have experienced. And we are seeing some really dramatic changes this year. And so with that warming, we're seeing increased hypoxia. It's also related to eutrophication of our coastal zones, and with that ocean acidification. And we're also seeing dramatic changes in the types of harmful algal blooms that are affecting us. And so one of the questions we're trying to explore is how and where are these factors interacting, and what are the results for ocean hazards. So in our region, we've got a long history of HABs, but mainly from paralytic shellfish poisoning, which is caused by this cell on the left. That kind of dark black circle is Alexandrium catenella. So this causes paralytic shellfish poisoning, and that has mainly impacted the coast of Maine in the late spring and early summer months. More recently, I'm not sure what's going on here. This must have an old recording. Sorry, it's a bad slide. More recently, we've started to see the emergence of amnesic shellfish poisoning caused by Syrenicea on the right, and also diuretic shellfish poisoning, another dinoflagellate, and then some aquaculture impacting organisms, Phrenia mikimotoi and Margolidinium polychrocortes. So these impacts are now year-round. They used to just be in the spring and summer. And so the way we've attempted to respond to this over the last several years is by deploying some advanced phytoplankton sensors across the region. The workhorse of this system is the imaging flow cytobot that you can see on the left here. This is an automated microscope that takes pictures of phytoplankton and other cells in the water continuously through deployments that can be up to 6 months. Then we use AI-based models to automatically classify these images into different species and genus categories. And then we complement that at several of our shoreside locations with another sensor called the environmental sample processor. This is sort of a molecular lab in a can. It can one of the things that's really helpful in our applications is that it can measure the concentration of domoic acid, which is a toxin produced by Pseudonecia diatoms. And the toxicity of those cells is widely variable, so it makes that measurement really, really valuable. So in launching this system, we focused initially on some established sensor installations with our colleagues at University of Rhode Island and here at WHOI, and then also up at Bowdoin College. And so we've established several of these and continued some others around the region, and we're aiming to actually expand this system up into the Maine Canadian border in the coming years in terms of our fixed locations. The other thing we've been working on, and Jamie gave a nice introduction to the power of these systems, is moving to some different mobile platforms. And so we have two kinds of mobility. We're using an ASV made by a company called C-Track that's based up in Marblehead, Massachusetts. This is essentially a floating solar panel. It has a really great capacity for supporting our sensors, which are relatively power hungry. And then we also have built some barge systems that we can kind of hopscotch around the region. They're set up to deploy and recover a mooring, and then we can trailer them to different locations along the coast. And so a big part of this effort also has been development of software for the integration of sensors on these different platforms, and that's something we've called PhytoArm. This is an open source software kit that's built using ROS, which is the robot operating system. It's essentially a type of middleware that facilitates interactions between sensors and servos and things like this. And we use it in a variety of ways on our barge platforms. We're mainly using it for interaction between our imaging flow cytobot, that automated microscope, and profiling CTD. So we can automate the collection of CTD profiles, and then we put the IFCB right where our chlorophyll fluorescence is most intense, and then we can really track blooms that vertically migrate in the water column. Another part of this effort has been the development of something called the HAB Hub, or WHOI HAB Hub. This is an interactive data visualization and access portal. It's been developed as an open source software project, so it's very easy for other folks to adapt or deploy their own version of it. We have one online that hosts all of the data that we're collecting through HAB on NE at habhub.hui.edu, and I invite you to check that out. And then Mindy Richland, my colleague here, has also made a nice set of tutorials about how to interact with the site at the Northeast HAB webpage. So kind of going back to some of the barge data that we collect, I'm thinking more specifically about HABs. So a lot of these cells are, particularly the toxin producers, are dinoflagellates, and many of these are able to swim in the water column, and many of them vertically migrate. So they go up near the surface during the day to optimize their exposure to sunlight, and then they go down the depth to improve their absorption of nutrients. And so this is interesting, and we're able to produce some really rich descriptions of these migrations through these deployments. And that's especially interesting in the context of acidified water, because in the coastal zone, OA is really complicated. This isn't a story about increased carbon dioxide concentrations in the atmosphere. It's more about the interaction with riverine inputs and the highly productive waters that are causing massive oscillations in carbonate system parameters. But where there is a really consistent signal is within some of these inshore systems where you have eutrophied water, a lot of productivity, Many of them have hypoxic bottom waters, and with that respiration is a lot of DIC and very low pH. And so those gradients in the carbonate are correlated with really strong gradients in nutrient concentrations that the cells are trying to access. And so one of the things that we're trying to tackle in the coming years is how do HAB cells in different species interact with these gradients through their vertical migrations. And so we've already, it's early days for us, but we've already seen some pretty significant differences between some of our HAB targets. So on the left is Alexandrium catenella in blue, outlined there. This is an analysis that my graduate student, Serena Sun-Clark, has done. That's where the pycnocline is in this inshore site we've been studying for many years. And then in black is the oxycline. So I meant to say also that oxygen is a nice proxy for kind of how these where the gradient lies for a lot of these parameters, but the specifics need to be worked out. So yeah, on the left is Alexandrium, on the right is Dinophysis, which is another really interesting HAB cell that causes diuretic shellfish poisoning. And this is one that doesn't migrate as much. It really kind of stays concentrated right at an oxycline in the water column. And so we're interested in exploring how that cell tolerates increased acidity in that area. So we do have some indications that these processes are relevant, at least, and perhaps driving what's been observed to be a really long-term trend in terms of increasing intensity of HABs. So in this NOSIT system where we have one of those systems deployed, Don Anderson and Judy Kleindienst here at WHOI have done some neat work developing an index to track the changing severity of HABs, and they've shown that this NOSIT system is one that has experienced a long-term increase in the intensity of its blooms, and that's particularly driven by earlier onset, which is down in the lower left, and also increasing duration of the blooms, and that seems to track pretty well with groundwater inputs and the rapid development of housing on Cape Cod here over the last 50 or 60 years. So there seems to be some link to nutrient pollution. And so with that, yeah, we're kind of digging more into this in the coming years. We have a new project with Alec Wong here at WHOI, Colin Rossler and Charles Culbertson up at Syracuse, and also our friends at Syracuse and Rubao G also here at WHOI, where we want to look at integrating two new sensors into these profiling systems. One is a high frequency carbon system sensor that Alec developed called Chanos 2, and then a multi-channel nutrient sensor that Colin and Charlie have acquired up in Maine. And so we're going to be using these systems to map in really high detail how cells are migrating and interacting with these different gradients in a couple of model systems. So that's about what I have to share today. I think the take homes from this presentation are first that the Gulf of Maine in particular is experiencing profound changes, profound climate changes, and that's affecting all ocean life, not just HABs. But we have these significant unknowns in the HAB world of where, when, and which HABs are going to be the most problematic as we look to the future. And so we don't have a ready answer for that. And so the best that we can do in the moment is to really build up our systems for surveillance and for monitoring these events when they do occur. And that's what we're doing through this HAB on NE project. And then the next sort of summary bits are just that warming, hypoxia, ocean acidification, HABs are all interrelated factors that are really driving outcomes in our ecosystems, among many others as well. And we are specifically interested in, as we see increased nutrient pollution and hypoxia-induced ocean acidification in the coastal areas around the region, how is that going to select for or maybe intensify certain HABs? And that's what I have. So lots of people involved in this work, and most of them are listed here, but not all of them. Thanks.
Thanks so much for that, Mike. That was a wonderful presentation. Up next, we have Adam Subhas, and Adam is an associate scientist at, Adam is an associate scientist at the Department of Marine Chemistry and Geochemistry at Woods Hole as well. He studies the marine carbon and alkalinity cycles with a focus on experiments and observations of carbon flows and transformations. Most of his work focuses on the intersection of chemistry and biology and how the ocean system cycles calcium carbonate minerals and carbon dioxide. In addition to open ocean research, he has several inshore projects, including some work in the Gulf of Maine and the Mississippi River plume in the Gulf of Mexico. All right, Adam, please feel free to share your screen and take it away.
All right, thank you, Austin. Let's give this a shot and see what happens here. Okay, I'm going to do this. So you're probably seeing, how about now? Is that good?
That looks perfect.
Excellent. So yeah, I'll just I'll give a shout out to Chris Hunt and Sam Sidlecki, who both I think conspired to invite me to this webinar series. So thank you both for that. I am pretty new to this group and to kind of regional ocean acidification monitoring. So I'm really interested to kind of engage with this group on some of the calcium carbonate work that we've done and see how it might apply to the Gulf of Maine and other places in the Northeast. So yeah, my focus is really on calcium carbonate and alkalinity cycling in the ocean. I've typically done most of my work in the open ocean, where most of the sort of surface reservoir of carbon resides, and that's shown here as this deep ocean inventory of dissolved inorganic carbon in gigatons. You can see that the deep ocean and upper ocean are by far the largest inventory of carbon on the surface of the planet. And, you know, this inventory is really there because of the ocean's ability to buffer its carbonate chemistry. And that's really driven by the really high levels of alkalinity in seawater. And so the alkalinity inventory and cycle is really kind of intimately tied to the carbon reservoir, and therefore how carbon moves from the atmosphere into the ocean and then where it goes from there. When I talk about alkalinity, you know, we kind of think about the basic end of the carbonate system. So bicarbonate, carbonate ions, plus, you know, you can kind of keep adding terms onto this equation if you want to. But really, you know, in terms of the cycling of alkalinity, it's really the calcium carbonate cycle that's driving a lot of this. So we think of, you know, corals and shellfish and other organisms on the coasts making calcium carbonate shells and skeletons. And out in the open ocean, we have organisms like foraminifera and coccolithophores and pteropods that are doing a majority of the open ocean calcification. Now, you know, the kind of interesting thing, the thing that I kind of study here is trying to put this all together and understand, you know, where is this alkalinity coming into the ocean? How does it get into the ocean? What happens in the ocean, both in terms of its cycling and then its ultimate burial in sediments, which is the main way that calcium carbonate or that alkalinity is removed from the whole ocean system. And so, you know, I think this is probably not all that new to this group, that we think about saturation states when we think about calcium carbonate cycling, and that saturation state, or omega, is defined as the calcium times carbonate ion product divided by the solubility of the mineral in question. And we typically think about precipitation happening in the surface ocean where this omega value is greater than 1, and then dissolution happening in the deep ocean where omega is less than 1. And of course, saturation states can be depressed both by the respiration of organic carbon, so really kind of intimately tying the organic carbon cycle to the calcium carbonate cycle. And also, of course, ocean acidification due to anthropogenic fossil fuel burning and ocean uptake. So calcium carbonate dissolution is kind of the main way that the ocean's going to restore alkalinity back to seawater. And this is kind of the main way that the ocean buffers itself against changes in CO2. So on this plot of total alkalinity versus dissolved inorganic carbon, I've contoured lines of constant PCO2 with the atmosphere being this red line here. When we respire organic matter, we kind of push ourselves up in DIC concentration. You could just as easily replace this with ocean acidification, right? We increase the partial pressure CO2. We decrease the saturation state, which then drives calcium carbonate dissolution. And that dissolution can actually bring us much closer to this atmospheric line here. So in terms of sort of the buffering capacity of the ocean, the calcium carbonate cycle is really kind of intimately tied to the ocean's ability to do this, this buffering. Now, interestingly, you know, a lot of my work has been in the upper ocean, focusing on in the open ocean, focusing on how upper ocean biogeochemistry interacts with this calcium carbonate cycle. So thinking about production, grazing, aggregate formation and export, what happens to sinking particles, and then ultimately what actually makes it to the seafloor and gets buried. This is where most of the action is in the organic carbon cycle, and it turns out that it's actually a really really active and kind of very dynamic part of the calcium carbonate cycle as well, despite the fact that we think of the ocean, the surface ocean, being super saturated, and therefore we wouldn't expect any dissolution of calcium carbonate to happen. So, you know, just to give you a perspective on this, there have been a number of different estimates of sort of the global production rate of calcium carbonate going back to kind of the 90s. And you'll see here that these numbers in general from the 90s going all the way up to this year are trending higher. So in the last sort of 20 to 30 years, we've started to realize that there's a lot more calcium carbonate production than we might have thought 20 or 30 years ago. So there's a lot happening. You know, it's climate relevant amounts. We're talking about gigatons of carbon. And only a fraction of that actually gets exported, which means that a significant portion of the calcium carbonate that's formed in the surface ocean gets dissolved, regardless of the fact that the surface ocean is supersaturated. And so this is a really important part of kind of understanding the dynamics of calcium carbonate cycling, and therefore the impact of calcium carbonate cycling on the alkalinity budget of the surface ocean and its ability to buffer the uptake of CO2. Now, just for some more sort of global perspective, we've kind of done some work both at Station Aloha and Ocean Station Papa, looking at sort of long-term sediment trap calcium carbonate fluxes. So this is a year day on the x-axis, and calcium carbonate export measured by sediment traps at Ocean Station Papa here on the left, up in the subpolar North Pacific, and then at the Hawaii Ocean Time Series down here in the oligotrophic waters of the subtropical North Pacific. And if we just kind of take measurements of the calcium carbonate inventory and then divide that by the kind of turnover time, we get production estimates that are much, much higher than what actually gets exported. So again, there's a lot of calcium carbonate recycling that is happening in the upper ocean, and this has implications for what's happening in terms of the alkalinity budget of these places as well. Now, I'm not going to go into the details of how we know this, but there's sort of the three main players of calcium carbonate producers in the ocean. So we have coccolithophores. This is actually a satellite image that was taken from, I believe, 2022 of the Gulf of Maine. So here's Cape Cod and WHOI, and we can see this just massive coccolithophore bloom from space. Obviously, paleoceanographers love to use foraminifera in their tests to do paleoceanography. And then we have pteropods that produce a more soluble form of calcium carbonate. And for a long time, people have thought that pteropods are really important. They're obviously sort of the canary in the coal mine when it comes to ocean acidification. But it's really coccolithophores that are doing a majority of the calcification and represent most of the inventory of calcium carbonate. And that's true even in the Gulf of Maine. I'll show some of that in just a bit. And this is I'll kind of focus on just one aspect of the things that we're learning about coccolithophore production and export. I got to participate in a cruise in 2021 on the Endeavor as part of this growing convergence research project with Rutgers and UNH. That's Liz Harvey at UNH, Kay Beidel at Rutgers, among some other folks. And over a kind of Lagrangian experiment, we kind of sat in the Wilkinson Basin and caught a coccolithophore bloom in action. So this is kind of the upper 30-meter PIC inventory over a roughly kind of 7 to 8 day period. And you can see it kind of coming up as the bloom initiates. We deployed sediment traps. So we have the export flux at the base of the mix layer, which are shown in these blue bars. You can see that kind of coming up along with the flux. And then the other thing that we were able to do was do incubations where we measured the production rate of calcium carbonate using a C13-based tracer. So this is very similar to what Barney Balch does with C14, but we're using carbon-13, which is the rare stable isotope of carbon. And I'll just make a pitch for this. C14 is great. It's incredibly sensitive. And we've learned a lot, but I think a lot of places are trying to move away from using radioactive isotopes. They're really kind of logistically difficult to deal with, and we're starting to get to the point where we can use carbon-13 based assays to do the same kind of experiments that have been done with carbon-14. So that's something to keep in mind, I think, as we move forward in terms of trying to measure production rates. But again, we can kind of take this inventory versus turnover time approach. We measure turnover time by the uptake of C13. We get a production timescale of roughly 10 to 12 days. That puts calcium carbonate production kind of way up here on this plot. And this is the amount getting exported, which means, even in a bloom in the Gulf of Maine, we're seeing something like 60% of that production getting dissolved in the upper part of the water column. And so this is a really dynamic system. And I think it's worth, this group thinking about, how we monitor calcium carbonate inventories, production, and export, especially as the Gulf of Maine kind of changes with time. Okay, I think I'm going to skip this, except to say that we're learning a lot about what's actually driving this dissolution. In terms of organic carbon primary production, a lot of the initial recycling actually happens by microingestion, by microzooplankton, and the digestion of that organic carbon by very small organisms. And the same thing is actually true of calcium carbonate dissolution. So we've done experiments in the lab where we label coccolithophores with carbon-13, feed those to microzooplankton, and then watch how much dissolves. This is work done by my graduate student, Chloe Dean, with really valuable input from Liz Harvey at UNH. This is the amount of calcium carbonate that's ingested, and this is the amount that actually these microzooplankton ingest, which means that something like 75 to 90% of the calcium carbonate that these microzooplankton eat gets dissolved in their guts. And so this is a really important part of the upper ocean calcium carbonate cycle. And actually, Mike, it'd be really interesting to know if you can resolve microzooplankton with the IFCB or something. Maybe we could get a sense of how these dynamics are playing out locally as well. That actually could be very interesting. I'll also make a plug for another recent cruise that I was kind of a shore-based scientist on, led by Kat Allen from the University of Maine. This cruise in August of 2021, they did a whole bunch of sediment coring all over the Gulf of Maine, including the Wilkinson Basin here and the Jordan Basin. These two cruises, or these two sites, we also have some results from a cruise that I led in April of that year. And we see some very interesting things in terms of what the profile of calcium carbonate concentrations look like at these sites. So in the Wilkinson Basin, we have most of the calcium carbonate actually as small particles defined as less than 53 microns. So this is predominantly coccolithophores, whereas there's much less calcium carbonate in the large particles, which is forams and pteropods. And we also see this really interesting increase in calcium carbonate concentrations near the deep. This is probably some sort of resuspension event that's happening that's worth kind of keeping in mind when we think about what's happening to this calcium carbonate. We see something very similar in the Jordan Basin up to the north, this kind of resuspended material that is in the deeper part of the water column. And actually, the highest concentrations of calcium carbonate are down here, which is not exactly what you'd expect to see. And we also have some thorium normalized fluxes that are coming in collaboration with Aaron Black up at the University of Rochester. So keep an eye out for that as those come out. So we think about, I was just kind of going back through the literature and trying to get a sense of what the calcium carbonate budget of the Gulf of Maine is. The most kind of extensive survey of what production rates are like, or actually a paper with Barney Balch from 2000, so almost 25 years ago now. They estimated a kind of annual average of about 30 milligrams of calcium carbonate per meter squared per day. This is our estimate in parentheses, which is within the kind of variance that they saw. Sediment trap export, the only data I could find is from a paper by Lloyd Kegwin and Cindy Pelskin, 2015. And then if we take kind of sedimentation rates and calcium carbonate percentages from CATS cores, we get this amount of surface sediment burial. And so what you can see is that burial is roughly, within error of what we think we can get right now, roughly 100% of what's getting exported, but it's only about 50% of the calcium carbonate that's getting produced. And so there's a lot of dissolution that's happening in the Gulf of Maine. And I think it'd be really good to get a handle on how this is impacting the carbonate system in the region. And I think the uncertainty in these numbers and just the fact that the best estimates of production go back to the early 2000s, I think speak to it's an argument for better constraints on PIC production and export in the Gulf of Maine. In terms of how we measure this, satellites are obviously a great resource. We're kind of interested in CubeSats potentially from Planet Labs or some other company that could provide really high resolution imagery, which would be really interesting. There's some sensors being developed that are a long ways off from actually deployment, as you can see from this like very simplified schematic. And this is a very recent paper that just came out describing this thing. And finally, we have lab-based techniques that can do this all. We can measure PIC concentration. We can do carbon isotopes. We can do this in a relatively high throughput manner, but we need physical samples, right? We need to get out and collect particles and then bring those back and measure them. So this is totally an option, especially if we wanted to pair and kind of vet some of these sensor-based approaches. But it does take actually getting out there and taking samples. I think I'm pretty much done, Austin. I'm looking at like almost time, so I'm gonna. Is that right? I'm like getting close to my 20
You have
minutes
about
here.
a minute if you do have like
Okay.
a summary slide.
Yeah. So I'll also just say, rivers are really important. I just took this figure from the NECAN website. Rivers are a really important part of the alkalinity budget and kind of seasonality in the Gulf of Maine. And it's worth considering, thinking about how these rivers are getting sampled and those implications for the alkalinity budget of the region. The other thing I'll just say really quickly is that we're also developing some kind of neural network approaches to doing carbonate chemistry in the region using available data products. And keep an eye out for Jenny Ruben's talk, who I think she's going to be speaking with you a little bit later in this series, talking about this work. So we're learning a lot about CO2 uptake and where this is happening, seasonality, things like that. So keep an eye out for that. So I'll just end there. I'm happy to take any questions from the panel with the other speakers. And thanks again for the invitation.
And thank you for that wonderful presentation, Adam. I'm now going to pass the floor over to Chris Hunt to facilitate the Q&A session.
Thanks, Austin. And thanks, everybody. That was great. And I appreciate you taking the time. So you may know it or you may not, but myself and the other people in the group have been talking behind your back. So we have a list of questions that we've been passing back and forth. So I'm going to kick it off, but I'm also going to invite the other members to jump in with their questions. But I wanted, since I'm had the baton right now, I wanted to start with Adam because I have a very specific in the weeds question for myself. Because I'm a total alkalinity obsessive, and so that obviously caught my attention. But so I'm really intrigued by the strength of the cycling of calcium carbonate in the surface ocean that you're finding, and it makes me think about the total alkalinity measurements that I make, but also about the tools that are commonly used to estimate total alkalinity from like other parameters, right? And so, how can we reconcile so much calcification dissolution going on in the surface ocean with the desire to try and estimate TA from whatever salinity or some combination of other parameters? And like, is that a bad idea? Right? I guess that's my big question. Is that a bad idea? Are we making a mistake there? Or do you think that the dissolution and calcification are kind of balanced out? And so you can still maybe get a handle on TA realistically over some sort of spatial temporal timescales?
Yeah, that's a great question. I'm actually interested to hear Jamie's thoughts on this as well. So, when it comes to alkalinity, I think what matters is the kind of, net export of calcium carbonate and what that means for, the removal of alkalinity from the surface. And obviously in regions like the Gulf of Maine, it's so seasonal and also so patchy in terms of, coccolithophore bloom is happening in one place and not another. And so I think those alkalinity salinity relationships are obviously going to change potentially seasonally. I know you and I have talked about this before. and are potentially going to be quite patchy as well. And so there's potentially an opportunity to, if you were to integrate some sort of optical backscatter sensor or something on a vehicle, and estimate what the PIC concentration is. It's not the same as a rate of production, but it might give you a better handle on how the calcium carbonate cycling is influencing your alkalinity salinity relationship. And I think, again, I think these things are going to be more stable out in the open ocean where things are just happening a little bit more slowly. But in the Gulf of Maine, it's going to be, I think, pretty dynamic.
And Jamie, I don't know if you want to jump in on that too. So I'm going to hijack things even further, but Adam's response kind of got me thinking about another question that I had for you, which is, and maybe this is opening a can of worms, but is there any concern about discrepancies on the sail drone specifically between making the PCO2 measurements, right at the surface layer and the rest of your measurements 2 meters below, right? I can see where in summertime where you have a super stratified surface layer, is there any risk there or any worries about that?
I guess I'll take that one first since now it's fresh.
Sure.
There is a sensor at like 50 centimeters and another at, I'm not gonna get it exactly right, but something like 2 meters or 3 meters. And we did mostly wintertime measurements and they were indistinguishable almost the whole time. And then as we got into the summer, because we did actually have summertime measurements that I haven't shown, as we got into the summer, there would be like 20-hour periods of low enough winds that they would be different. And but they were very short. They were very noticeable. Like we said, ooh, look at the difference, and
sure.
So just very few instances. I don't have a good answer for the salinity total alkalinity question because, well, maybe because I'm not totally understanding. If you're studying alkalinity, like I guess you'd want to measure alkalinity, right? And maybe even plotting it as an anomaly relative to the expected alkalinity from the STA relationship would be really useful for things. And I can imagine scenarios where that would be the case. Like if you're wanting to look at the riverine input or its seasonality or some anomaly from biology, you'd really want to have some direct measurement and then look at its anomaly from the expected value would be really useful. But if you were interested in the carbon system and the errors were immaterial to the question you were asking, like DIC accumulation over a 10-year period, then I would guess that would be good enough. And so I guess that's why I put it up there. It just, I guess it depends on the question. I don't know. I'm not familiar enough with the questions that are being asked to know, but I'm guessing Adam's really interesting ones about PIC. You want real measurements, not just relationships.
Yeah, I think it's also, if you want to talk about like omega, for instance, right? If omega is one of your metrics of ocean acidification, then how sensitive is omega to TA? Maybe that it's somewhat insensitive, right? I've played around with some of those numbers and TA makes some difference, but it's not as much as I expected, quite honestly. So maybe there's some advantage in using TA just because the sensitivity to omega is less, if omega is something that you're really concerned about. Let's see, I don't know, Beth or Kamiko, I don't know if either one of you would like to jump in on one of your questions. You have some really nice ones.
Sorry, Adam, I have a question about the budget. How is the lateral transport when you do not just the vertical? The reason why I'm asking is we have a strong coccolith plume of Grand Banks, but we don't have any calcium carbonate sediment at the bottom. It's really transferred away. So alkalinity removal from the region that way. I assume Gulf of Maine is much more confined, but
yeah, it's a really good question. That's one of the things we're hoping to learn from the thorium-based fluxes, is get an estimate of how much sort of sediment focusing is happening. The Gulf of Maine is, I think, fairly well balanced in terms of sediment inputs. Like it's because of the bathymetry I think a lot less sediment is lost out of the region. And I know Matt Charette did a budget for organic carbon that looked like it was pretty well balanced. But again, these are fairly old numbers. I don't know if anyone has really revisited this in detail in some time. So, and I'll just point out that you know, these sediments in the Gulf of Maine are only like 2% calcium carbonate, and so they're really high sedimentation rate, but not a whole lot of calcium carbonate is actually getting buried. And so you're going to end up being quite sensitive to the dilution from other sedimentary inputs. And that's a pretty big uncertainty. I would say that's another reason why kind of getting a better handle on that and what the variability is like would be really important. And again, I think there was only the one paper that I found that had kind of annual sediment trap data in terms of the export fluxes. And so I think there's just an opportunity to increase those observations and get a better sense of what's going on. But yeah, I would say that where the sediment goes is going to be a really important part of the alkalinity budget for sure.
And the other question is, you showed the calcium carbonate, the export rate is production rate increase over time. What were we missing? Like reminds me that heavy metal concentration in the ocean increases over time because we reduce the contamination. What we are missing before?
Yeah, I would say it's better observations and just a more focus on it. I think people had always assumed that because the surface ocean is super saturated, that whatever is produced is exported. So people would kind of assume that the export rate is the same as what's getting produced above, but we're just finding that to not be true. And what that leaves is this imbalance between what we're actually measuring as production and what's getting exported, and that has to be redissolved and recycled in the upper ocean. So yeah, that, you know, kind of like Jamie was showing, right? The more observations are leading to, you know, a larger gap between models and observations. I think the same thing is true. The more we look into the calcium carbonate cycle, the more we're realizing it's like very heavily recycled.
Thank you.
Yes, do you want to go ahead?
Yeah, thanks. I had a couple of questions for Mike. So I have heard about, you know, all of your Hab on and the Hab hub and everything. Do you think that there's any utility to have OA parameters added to the Hab hub? In other words, do any of your stakeholders really care about OA? I mean, they're very focused on shellfish safety, but are they interested at all in OA impacts to shellfish?
I think potentially it's definitely a focus area for the new project is to engage some stakeholder communities about types of OA products, OA specific products they might like to see. Our observational footprint in the new project is pretty limited in scope. So we're really focused on these inshore systems that are tractable for understanding these interactions between the cell's behavior and these sharp gradients and carbonate system parameters. But it's certainly a place to start in terms of prototyping some ideas about what people might like to see in an interactive portal.
Yeah, we'll be really interested to kind of go along with you and your project and better understand those interactions and what people really want to know about it. Because I think that's going to be really interesting as that goes forward. And then more specifically about the HABs themselves, you showed data that had different vertical migration rates for different species. Are there any theoretical bases for thinking that OA would affect some of these species more than others?
Yeah, I think, well, one of the things that I'm particularly interested in is with dinoflagellates. So dinoflagellates have a wide range of sizes, and so with that, they have different half saturation constants for absorption of nutrients. And so the sort of optimal ambient nutrient concentration for efficient absorption may change between species. I think the other thing that is of interest, but not where we're starting, is how this sort of maps onto populations that are developing offshore in coastal zones where there's been documented vertical migrations of tens of meters. And so that's beyond a diel period that cells can actually traverse in a single day, but may be interesting for understanding different selective pressures across a community. So not just the HAB species, but maybe some smaller. And, like this year, the story in the Gulf of Maine is this tripos bloom, which is a significantly larger dinoflagellate.
Yeah, Mike, that ties into a question that I had is, are the HAB observations targeted to species that produce toxins? And this is me speaking as a really bad biologist, or a person who doesn't know biology. So are your observations really focused on certain species, or are you capturing kind of the whole community as you make your observations? And then could those observations be applied to, like you said, like the tripos bloom this year, or some other community that may not specifically be a HAB species, but could have an effect on the OA scenario, at least seasonally, like we're seeing?
Yeah, so we're limited by our tool, as everyone is. The IFCB technology is really focused on capturing, it's specified to capture cells between 10 and 150 microns in length. It's not a great EHOC sensor, so back to Adam's question. And it's also not, we don't think it's a particularly good sensor for the tripos blooms either. So tripos is sort of on the too big end of that range, and the EHOCs tend to be too small. We do sort of within that size range, though, we do a good job of capturing the community. Depending on where we have the sensors, we will either trigger on cells that have chlorophyll. So that obviously is biasing towards photosynthetic organisms. But when we're offshore, we do sample or we detect cells based on scatter. And so that can be a nice way to capture some of the micrograzer community that Adam talked about. And so we do see that in a lot of our data sets. So, that's and the IFCB is really the lion's share of our observations. So we have these ESP sensors. Just by sheer volume of numbers and observations, we have a lot less of the ESP observations, but it's a great tool for specifically detecting the toxins. And so we also have some questions about interactions between physical chemical conditions in the ocean and dynamics within Pseudonychia blooms. So selection for different species, changes in toxicity, et cetera.
Awesome. I see Kamika has her hand up, and then I see Adam Paventa too.
I think Adam raised his hands first, so maybe he can go first.
How polite.
All right, so this is a question for Adam. Does the surface dissolution of calcium carbonate that you saw show up in changes in the total size of the DIC pool, or can you detect, any changes in the speciation of the DIC pool, like a measurable increase in carbonate?
Yeah, that's a really good question. So Chris and I have chatted about this, that it's possible that sort of seasonal changes in that alkalinity to salinity relationship are due to coccolithopore blooms and sort of hotspots in calcification. So we've kind of made attempts to close the alkalinity budget using this kind of upper ocean dissolution out in the open ocean. And I think it'd be really interesting to start looking at that in these more coastal areas like the Gulf of Maine to really understand the impact on the carbonate system. Because ultimately, if you're removing a bunch of alkalinity via calcification, you're going to really reduce the buffering capacity of that seawater. And, the buffering capacity of the Gulf of Maine is already quite low. And so it's kind of a positive feedback on that. And so understanding those dynamics and its patchiness and seasonality, kind of all that stuff, I see as potentially being important for regional OA monitoring.
All right, great. Thank you.
Yeah.
I have a question to Jamie. For how long you need for lead time to hire a sail drone? It sounds really fantastic to detect something you find in satellite or something offshore, coastal area to the shelf. But you can't really mobilize your ship right away, right? Especially big ships we have. But a sail drone would be perfect to set it out. But how long you need to book ahead of time or something?
Oh, that's an interesting question. You're right that it's faster. They have a big fleet, ready to go. So, but I would guess the real bottleneck would be who's funding it, right? How do you get the funding to flow to get to sail drone to get them out the door? So if you had some way of having like a contract with them, then I think it could be very fast. So NOAA has a hurricane watching sail drones, and they have them kind of at the ready on the Atlantic coast, like Florida, and there's a Petersburg office. And so St. Petersburg. And so I think they send them out, or they have them in the hurricane zone, and then they dash this way and that way, trying to get underneath the hurricanes. These have the shorter wings, so they're more robust to high waves. So I think the bottleneck isn't sail drones mobilization, it's all the other pieces that lead to mobilization. So maybe if you had a contract for fast mobilization, they had one staged somewhere, then the pilots would be ready to go. I would guess. I've never been in that exact situation.
Yeah, as I said, there is a cockroach bloom of green Grand Banks, but we never been there. We only saw it in satellite. So if we contract 10K or something ahead of time, and it's okay, it's starting, send it out. Is that possible?
They are not so interested in doing 10K would be 2 days of 2 sail drones. They would never.
Right. Yeah, that's true.
No.
So yeah. I have to put more money in it, but
much more. So yeah, that's interesting. So we actually were pretty close. We kind of touched the tail of the Grand Banks. That's a region I'm been kind of obsessed with for lots of reasons. So we were over there a little bit in the winter and spring. So maybe we should talk about what you're looking for, and did you say there's a bloom? Is that what you were interested in?
Yeah, so usually late summer, right? And funny enough, you see that sort of a satellite signal of PIC looking stuff in wintertime by resuspension. It's not that deep. So my guess is settled during the fall time after bloom, but it resuspended during winter time with a huge storm and washed away somewhere.
Okay, maybe we can talk sometime afterwards. I'd be curious what you'd be looking for, and if we weren't there that much, and definitely not in summer, but we were over in that area for a little bit in the winter. So definitely during some strong weather. So
Amy, can I follow up with the question? Oh, sorry. Who was
that was me, Chris. I was just going to say, Kamiko, if you're interested in, so we have a contract with Planet Labs that does high resolution satellite imagery, and they it's sub-meter resolution. It's really high. And so if you're interested in something like that, you can get a contract for about $15,000, which gives you kind of on-demand tasking. And it's 24 hours advance notice. You can book an image of basically anywhere along the coast. And so that might be a nice way to at least kind of look in a little bit more detail.
Yeah, we looked at last 10, 20 years satellite every month. And funny enough, the cockroach sort of a signal is not every year there, but often there. But we are sort of amazed how strong wintertime signal is.
Interesting.
So resuspension seems to be happening quite a lot.
Yeah, are there wavelengths similar to resuspended material?
I had a Kokoris scientist before, and
Yeah.
he was looking through it, and
Yeah.
he thought that was actually Kokoris signal.
Okay. Huh. Because I feel like around here, the times where we've seen cocculus, we think, have been in the summertime in general. And they're really unpredictable, so it's hard to tell.
Yeah, usually late summer.
Mhm.
And I thought Kokoris bloom is something to do with water stratification and depletion of the nutrients after the diatom bloom. So usually late summer, and we never had a cruise around that time. So
Well, Jamie, can I ask you one more question too, which was I was interested in the neural net results that you showed from sort of predicting the PCO2. So, like, if you had a magic wand, how much sail drone data would it take to apply that neural net approach sort of on like a regional scale or like on a broad scale? Or would you even want to do that?
I don't think I know the answer to that question, but when you say regional, how
Well, let's just say the Gulf of Maine.
Gulf of Maine?
Yeah. Right.
I think, I don't know, I think we need to do a lot more work, like to
Okay.
take it and degrade it and see what you'd need to do it. I actually think I'd really be interested in seeing like a whole spin up of an approach where you'd do this in a you'd sample a model like you would
sure.
before you go start putting expensive instruments in the ocean, sample the model as you would with a sail drone, then reconstruct the model field, with the output using these fancy techniques, then degrade the model output and do that iteratively until you kind of come up with a parsimonious sampling plan, then go in the ocean and do it and see how well it worked. But right now we just did the one experiment. I don't know. I don't have any truth to compare it to, so I don't really know.
Right. And you need that model, first of all, which is still kind of
and at high enough resolution to make sense to even do this.
Right. Yeah. Because the 1 degree by 1 degree is probably not going to get you that much in the Gulf of Maine. So
Nothing like that. Nothing like 1 degree. Something that it doesn't have to be right, but it has to have the kind of right variance.
Sure.
And resolution commensurate to the process you want to study. And then you can see if your statistics of the reconstruction match the statistics of the model truth, where that model truth at least has the right variance, spatial and temporal.
I know we're starting to butt up against our designated time for question and answers. I didn't know if anybody else from the audience had anything they wanted to ask. I have one more that I would throw out there, but I also don't want to take up everybody's time. I'm going to say no.
Go ahead, Chris.
Okay, all right. My last one, this may be easier, maybe not, but it's also for Jamie. Has anybody tried to put any other carbonate system sensors on sail drone besides PCO2? I mean, you mentioned pH, but has anybody tried TA or DIC?
I'm pretty sure the answer is no. Not to my knowledge, it hasn't happened, but I don't. Yeah,
that was my expectation, but I didn't know if somebody out there had tried it.
Yeah. I've heard of a DIC sensor out of Southampton, but I don't know what the status is. I think they're trying to put it on gliders, would be my guess.
Yeah.
But yeah, we're potentially going to have some of the BGC Argo sensor packages on gliders soon in the Gulf of Maine, but that's also a few years away.
Yeah, and I've been hearing rumbles that the pH sensors for the gliders are going to be kind of extremely hard to get, at least for a little while. Yeah. But I just today got this paper, but somebody's put together an in situ TA sensor that looks really interesting. And I know Alex DIC system is also really interesting, but maybe not set up to integrate with something like a sail drone. But there's definitely like a sort of scientific need for something like that on a platform like a sail drone or a glider. So we'll keep looking, I guess.
Yeah, it seems like groups are trying. There's Lab on a Chip in the UK.
Yeah,
They've tried to do
right.
that, and then like I said, in Alaska, they've had some promising results, but I don't think it's DIC or TA. I think it's still PCO2.
Yeah. Yeah, there's a Lab on the Chip TA out of Canada that's promising, but it's still like looks pretty bulky, and the number of measurements you get out of it is on the low side. So I
Yeah.
think that's still
And
like
the ones
Yeah, I was
that-
told it's a little long way to go still.
Yeah, it seems that way.
yeah.
Yeah.
I'm curious, actually, if you could comment on whether or not you were involved in kind of pushing to update the sensor payload and package on the sail drone platform. That, you know, that's a frustration on our side. It's a really capable platform, but it's not one that seems to be immediately accessible for like a can that takes lots of pictures. So I wonder, you know, I wonder how much you explored that and if you had success getting them to update the sensor payload.
Oh, no, we didn't. The only thing I pushed for is to reintegrate pH, but without success. So I don't know if that's, yeah, it's a company and they're trying to like do the most efficient thing, but are you looking for an underwater camera? Is that what you're saying? Some kind of video?
Well, I mean, what we use a different platform. It's a much more open-ended one. Our use case is not as complex as yours. We're trying to actually use the system more as like a cheap buoy. We just have it go out and stay in one place. And that way we don't have to pay a lot of operator costs. We just have it hold station. We just finished a month-long deployment. It was pretty successful. So, and the advantage on our side, it's not as large a vessel, but it has a lot of power. You know, we can have 50 watts continuous essentially running our instrumentation, which is enough for us to do a lot of interesting things. I don't know how that compares with the sail drone, but to the extent we've had a chance to explore it, we've kind of hit a roadblock immediately that, you know, if they don't offer the data as a service, then there's not a lot more to talk about.
Yeah. The power is an issue. Like just the power, one of the most power hungry ones is a radiometer. And we just, and it's not even, the data quality is so, we just turned it off. Like, so it's on there, but we turned it off. So because it wasn't giving us anything we needed. So I'm guessing that they're looking at maybe the sensor you're thinking and just thinking we won't be able to, right? They want to be out as long duration. They want to be out for months and keeping it going.
Are you using the smallest version or using one of the larger boats that they offer?
I think it's called the Explorer that we have, and they have a huge one that we have not used. So we have like the 10-foot, the 10-meter wing, the 5-meter keel, and they have a much bigger one that we've never used. Yeah, but those, I would guess the price on that would be insane. And I don't know, getting that out in the coastal waters probably not super easy. So. I think they have a commercial mandate, right? It's a company. And I think they're doing a lot of maritime awareness and defense contract now. And we're kind of keeping them having a kind of science mission, but I think they see that as a side thing now. I'm talking out of turn really, but we're not making them a lot of money. So but they have a really powerful platform and really excellent pilots and amazing data. So I think I don't know, I'm willing to kind of work around those constraints for the things that I've been working on, but it's not endlessly versatile in that sense.
I'm gonna jump in here, and as we are now 6 minutes over, I'm going to close out the webinar by just giving a big thank you to our 3 presenters today, and thank you for everyone else for being able to stick around for a couple minutes at the end. I want to remind everyone that our next webinar coming up is on August 23rd at 1 to 2:30 once again, and that will be focusing on Indigenous interests, concerns, and perspectives. If you're looking to rewatch this webinar, it will be on the NECAN website. And with that, I'd like to thank everyone again, and we'll see you in just under a month at our next webinar. Thank you.